Hi, everyone. Welcome back to A4LI Scientific Spotlight. I'm your host, Brenda Eep. Today, we have Alex Trapp from Retro Biosciences. He's a computational biologist who's passionate about bridging his work with experimental biologists to find ways to tackle the hallmarks of aging through rejuvenation. Continue listening to learn how it all began. Thank you, Alex, for joining us today.
Thank you. It's great to be here.
So how I like to start these conversations is by asking you, I guess, where you're originally from and what initially sparked your interest in science.
Ooh. A bit of a long journey for me. So I was born in the south of France,
Mhm.
Toulouse, although my family's from the northeast of France. Spent some time in Germany when I was younger as well. Lived in Paris for a bit. And then when I was 9, we moved to the US. We moved to Dallas, Texas. And that was a big, big culture shock moving from Europe to the US. I learned English along the way. We also lived in the Buffalo, New York region as well. And then I ended up going to school, to college at University of Rochester in Western New York, which was really dope. And I would say that's really kind of where I was interested in science beforehand as well. I was really interested in biology in high school, but I think it's really when you're exposed to kind of higher education in college that you can really get interested in it. And for me, that was through research. So it's really cool to read a textbook and to learn about what other people have discovered. And it of course, builds your foundations for being a scientist. But to be able to make those discoveries yourself, I think is just like infinitely more powerful. So I would say that. Uh,
Yeah. Yeah. I was recently told about I guess there's a geroscience effort too in Toulouse, right? And there's an
correct.
meeting that happens too, and it's a I think it's a great conference too. I think the previous one was on clinical trials, and I just heard that it was a very collaborative environment from everyone around the world interested and engaged in the field. So it's really good to see.
Yeah.
Um, Wow, you've been everywhere. I mean, definitely a culture shock, especially in Texas. I,
Yeah.
um,
Paris, Texas was a fun one.
yeah. I mean, that's really interesting. I mean, so do you have any family members in STEM, or was it just the college exposure and biology that interested you?
No one in my extended family is into biological research, but I have a couple of family members who are like healthcare professionals. So my grandparents were both psychiatric nurses in France,
Mhm.
and my uncle is a psychiatrist or was a psychiatrist until he retired. And I have two cousins, one who's training to be a psychiatrist and one for ophthalmology. But yeah, no one. I mean, my parents are more in the my mother and father studied economics and finance at university. So definitely more kind of oriented towards there. So kind of, yeah, the
Well, I
black
mean,
sheep.
aging,
But
aging and economics. There's
a
a lot
for
of white
sure.
yeah, a lot of white papers on how it affects our economy and stuff like that. So
Yeah.
definitely
Yeah.
super interesting. And in your family too, like your cousin, I think your cousin ophthalmologist. I think there's a lot of eye research going on too. So super
Yeah,
interesting.
like Life Biosciences, David Sinclair's company.
Yeah,
Yeah, excited to see what they put out.
yeah, it's a collaborative effort in the end.
Indeed.
So you did your, I think, bachelor's at Rochester, right? With
Yeah.
Vera,
Yeah,
right?
I worked with Vera Gorbunova and Andrei Silvanov, and David Goldfarb, who was my first kind of mentor in biology. He was more in like the nuclear transport space, but he was getting really into aging towards the end of his career. Um,
Mhm.
I was emeritus, but he was really into yeast aging. And I thought that was like he was like modulating it like incredibly. And I was like, wow, I didn't know you could do that for lifespan. And so yeah, then he put me in touch with Vera and Stefan in her lab and started studying naked Morax. And I was there for 2.5 years. Started on the experimental side and then slowly transitioned over to the computational side. I spent a couple months in London studying abroad at UCL and took my first computational biology class there. And that was an eye opener. I just thought I could contribute like a lot more to that space than the experimental side. But I really value kind of the hybrid
Yeah.
um, environment that I trained in.
Yeah, I saw the recent news with the Matrix Biosciences, right? The Pfizer company. Did you have anything involved with the hydrolonic acid finding and the naked mole rats in Vera's lab? Or
Yeah, so not myself. It was a little bit earlier. It was in 2013. There was a Nature paper. First author was Zhao Tian, who's just starting his lab in San Diego now. He spent time with David Sinclair after Vera's lab.
oh,
Um, but that was really cool work. I did work with Nick and more at stem cells. They don't produce much hyaluronic acid. At least the fibroblasts mostly do, and that's what was studied in this 2013 Nature paper. But yeah, in my case, I worked with the hematopoietic stem cells and trying to characterize the immune system, along with the postdoc that I was working with. And then also the mesenchymal stem cells, so the niche around the hematopoietic stem cells and how kind of it all comes together to make this animal kind of remarkably long-lived and resistant to many diseases.
yeah, it's interesting. I know a lot of people or a lot of companies uh, went towards the naked mole rat avenue, right? Because mice, they live long because they get the cush life in a cage, right? And the good hygiene and everything. So how did you find yourself at Dr. Gladishev's lab doing computation bio?
Yes, I graduated college in 2020, right as the pandemic hit. I was originally kind of more interested in pursuing, at the time, a double degree, an MD PhD.
Oh,
I thought I could contribute a lot to humanity that way. And then I realized that just medicine was not for me. So I was headed more into the research track. But before starting a PhD, I just kind of wanted to get more research experience. And especially because I had made this shift from experimental to computational biology. I had done a little bit in undergrad and taking a couple courses, but I think you really learn when you're just like fully ingrained in the computational team and just like grinding out a problem
yeah.
and kind of learning by yourself. So yeah, I emailed Vadim, I remember, a couple of months before graduating, and I was like, "Hey man, it'd be awesome if I could pull through and do some cool work with you." And it happened that was the case. I started working kind of experimentally with a postdoc there at the time, now professor in Barcelona. His name is Marco.
Mhm.
Because Vadim was originally interested in selenoproteins, so he transitioned over to aging in the second part of his career. But in the first part of his career, he kind of became the guy to study selenoproteins.
Mhm.
And there's only a few in the human proteome, but they're very important for a variety of redox reactions. And if you don't have these selenoproteins, you die.
Yeah.
And the way that they're made is that there's a 21st amino acid, selenocysteine, which is not normally encoded by codons, so sets of 3 nucleotides, but it's instead encoded by stop codon, which normally signifies termination of the amino acid sequence. And it's dependent on a 3' element called the second element. So it's just like 3D organization that, like, if you have this particular element after this stop codon, you will read through the stop codon and instead insert selenocysteine as opposed to the stop. So we were interested in working on this and maybe do some gene therapies or something like that. Very proof of concept. But yeah, eventually I switched over to a more computational project because Vadim was really interested in this single cell epigenetic clock landscape, which I guess is kind of where I blossomed as a
Yeah.
scientist in some ways. It was a really hard problem. People had been I'm sure people that are viewing this are semi-familiar with epigenetic clocks and the concept of tracking biological age, but all this had been done in the bulk setting beforehand, which means that you're getting it from like millions of blood cells at a time, or maybe a sample of liver from a mouse, right? And you're just looking at like the average. And that's interesting, right? You can tell maybe the average of this after a treatment, maybe the average goes down, which is still useful, but you're really not getting that I don't know, the cellular resolution that you would need to see because
Mhm.
maybe some cells are getting younger and some cells are not. Some cells are getting older, for example. And so being able to measure aging at the level of single cells, especially in interventions that are highly heterogeneous, I think is really important. And so worked on that for a while.
Yeah, and I liked your 2021 paper too. I think that kind of made it for you, right?
Yeah, it was a good paper. We were fortunate enough to get the cover of Nature Aging in the 12th issue of the first volume. So that because it started the same year, so it
Yeah.
happened in December. Yeah, that was really cool. Vadim is a fantastic mentor, and he just thinks about aging in a really good way, and kind of trying to get at these very fundamental questions of when does it begin, why does it happen, are the changes that we observe damaging or protective? All these things I think are super interesting. So Yeah, I also think I'll just mention two other things from that time, maybe three, but uh, one really interesting finding that we had, and this was a finding of my colleague Csaba, who's now a professor in Hungary, is that there's this rejuvenation event during embryogenesis, which I think is like really critical because so many companies now are pursuing reprogramming or rejuvenation therapies using fairly artificial means, right? That people discovered in the 2000s and later won Nobel Prize for. But the fact that our body does it every generation, right? Because the sex cells, the sperm, and the oocyte, they age with you during your life, but
Yeah.
somehow they need to give rise to an embryo that is always age zero, or every year, or every time you have a child. And otherwise, every generation, people would just get increasingly older.
Mhm.
So kind of applying epigenetic clocks to this like very early part of embryonic development and really uncovering this like decrease in epigenetic age during gastrulation when you have a lot of cell division, and then just kind of a monotonic increase and kind of finding what seems to be the start of the aging process, which really seems to happen in development.
Yeah, I mean, my colleague Carlos, right? He kind of extended that idea at the hypothesis prize at Foresight too. And
Yeah,
right,
I saw that. I was there when that happened.
yeah,
Yeah, it was really nice. I
yeah,
think it's a really promising area of research. But a lot of work just needs to be done because we really have no idea what happens. Like there's a lot of papers that describe embryonic development, but it's such a complex process and really kind of harnessing the parts that matter for us while not kind of resetting the entire epigenome to where the cell becomes pluripotent.
yeah. I heard the African, not the African killifish, just I think zebrafish are really good organisms to study embryogenesis. But I'm sure there are other organisms that are best suited for that study too. Okay,
Yeah,
so let's see. So you did some research, right? And Dr. Glavish's lab, right? And then were you one of the first hires at Retro?
indeed. Yeah.
Ah, cool.
Yeah, the company started, I think almost like officially almost 3 years ago, but we really like acquired lab space and got things going in the fall of 2020, or yeah, like the winter of 2021, basically, when my paper, around the time that my paper came out.
Yeah,
I met Joe, who's the CEO, remarkable individual, at a Foresight Vision Weekend in 2021.
nice.
Yeah,
Yeah,
I was introduced to him by a couple of people, and then I was really on the track of like the very, I don't know, like what is expected of you kind of when you go into science. Are you going to do a PhD? You're going to do a postdoc? You're going to become faculty? And like that was an appealing path, but at the same time, it just takes so long. I just wanted to like really get into the action
yeah,
and really contribute to making medicines. Like I really enjoyed like publishing papers, and it's great to talk about them and to make the occasional tweets or give a conference talk, but to like actually translate a finding of a paper, whether it's methodological or like therapeutic, to the clinic, right, into like actually like human beings. You receive a treatment that you helped to create, and it somehow helps them, right? Whether it's extending their life or even just curing some kind of disease. I think it's like perhaps the most powerful thing that I can be doing. And so that's kind of why I switched over to industry and been at Retro now for almost 2 years, I guess. And it's been a great honor.
yeah. So yeah, I was actually pretty excited when Joe mentioned that Retro was starting a graduate research fellowship, right? So like that kind of exposure that you got in a lab, but you can do that at Retro, which is amazing, and kind of figure it out on your own there too, right? Become a independent scientist there without any obligations, like ties to like a specific program, whether it's a master's or a PhD. So it's super awesome.
It's still early days for that program. We only have a couple of people in it at the moment, but yeah, I think it's a great alternative to kind of be able to provide the experiences that I was able to get, like you mentioned, in academia, but like all enclosed for people who are just like really, really talented, but perhaps only graduating college, right? And they don't have like the classic like PhD and postdoc experience that most biotechs.
Yeah, exactly. So what are you building that Retro?
Yeah, we're building a bunch of things. 5 programs at the moment. One program is interested in plasma protein modulation. So there's evidence from a variety of papers and also some of the parabiosis literature, which I was also involved in when I was in Vadim's lab. We published a nice paper this year that shows that parabiosis effects are quite durable, and that you do rejuvenate the old parabiosis partner. This is when you connect a young and an old mouse together. Um, and so it's kind of inspired by the findings from this, and it's trying to see if we can make any kind of translational intervention that somehow modulates the blood profile to where perhaps you remove some of the bad factors that are accumulating in older people, or maybe you would add some youthful factors that young people have more of. And then we have an autophagy program, which is mostly just a small molecule that we're pushing through the clinical trials. I don't interface with that program too much. They don't generate a huge amount of sequencing data. I'm a computational biologist, so I'm really into sequencing data. But I think what I'm most bullish about and most passionate for, like, long-term effect sizes is really, and it's just like really, large effect sizes as well, is reprogramming. And so right now we have 3 subsets of this like broader reprogramming division. We're interested in T-cell reprogramming. I think this makes the most sense in the context of immunotherapy to begin with. So T cells naturally, when they fight cancer and when they get older, and in older people, they just have decreased efficacy at fighting the cancer.
Yeah.
And if you were able to, for example, harness the T cells that are infiltrating into a tumor, but are perhaps exhausted because the tumor microenvironment is making them so that they have no effect on the tumor, but they do have antigen specificity for it. So they are seeing it. If you were to extract those T cells and then perhaps reprogram them to a more naive state, that would permit them to attack the tumor as opposed to when they were in the exhausted state, and then expand them and put them back into the patient, you could perhaps cure some solid tumors. And so we're really interested in that paradigm, cell tumor infiltrating lymphocytes, but also exploring other avenues within the T cell space. It's just really tractable because you can just get them. Ideally, you get your T cells from a blood transfusion that you would just get. A lot of the in vivo stuff is much harder, right? Like you mentioned the eye earlier in our conversation, that you have to deliver something into a human or to an animal.
Yeah.
It's like complex, but we are also interested in that. So we have a program in liver reprogramming. So kind of trying to piece apart what happens during in vivo reprogramming and the right delivery methods and what the right dosages are, and maybe if you need cycling or not, just exploring that generally. And then our last program, which I'm also really interested in, is our HSC reprogramming program, so hematopoietic stem cells. So it's kind of neat because I worked on these cells in naked rats at the beginning of my scientific career, and so I'm working on them again now. And the idea there is that there's been a couple of papers that show that transplantation of young hematopoietic stem cells into old seems to have beneficial effects. And so we're just kind of really interested in moving that forward. And instead of having to have a source of young HSCs that you just mine from for other people, you could do this in an autologous fashion, right? So where you take an older person's hematopoietic stem cells and then you rejuvenate them and then put them back into the body. So
yeah, it's amazing. I think the first time I learned about iPSCs too, I was like, oh wow, you can take these from patients and correct them and then give them back. So there's less chance of, like an invasion, like a threat to the immune system, because it's from a familiar, it's from their fluids, right? Their body. So it's more recognizable.
Yeah. Autologous is nice in that regard, although it's also much more logistically complex because you need to get the cells in the first place, bring them over at some point. Maybe we'll have like a manufacturing cell therapy manufacturing facility. So you need to get them over to this facility, do all your things, and then put them back into the patient. And especially in the case of hematopoietic stem cells, we're developing methods to condition the bone marrow. So normally when you do these hematopoietic or bone marrow transplantations, you need to get rid of the old bone marrow because if there's competition between both, it's not good. And getting rid of this old bone marrow is usually very toxic. People use chemotherapy, radiation in most cases, and we're developing methods to do it in a non-toxic way so that it could be amenable for just like broader applications. But again, still very early in the research phase, but really cool data coming from that. I guess I can introduce what I do as well. So I lead the computational team there. People, it's really cool. We just work with like so many different modalities of data, from imaging to single cell RNA-seq, recent forays into flow cytometry, which gives you much quicker readouts as opposed to single cell, where you need to make libraries and sequence them. Just multimodal, trying to understand aging the highest resolution that we can, and as many different kind of perspectives as we can. And hopefully that gives us some hints as to how to rejuvenate organisms.
Yeah. So, I mean, I like to ask people who are scientists, how do they describe their work to their friends or family who don't have a science background? How do you start that off?
Yeah, it's an interesting question. I guess that's the case for me because most of my family doesn't have a science background. I guess a lot of friends in college as well didn't. Yeah, for me, it's a lot of, I mean, ever since I transitioned more into computational biology, it's obviously a lot of coding. So, yeah, creating algorithms, using existing algorithms of some kind to analyze your data sets, thinking about effective visualizations and how you want to communicate the results to wet lab scientists. There can be a disconnect, right, that happens between what happens in the computational space and an experimental space. It's like very tangible experimentally, but then an experimentalist might look at some plots and really have no idea how they were generated, right? So making sure that the communication is really effective. And yeah, just generally kind of also like keeping up with literature is crucial. So reading a lot of papers, especially since we have multiple programs and our
Yeah.
particular kind of supportive program, computational biology, interfaces with all of them. So you need to kind of be at the state of the art of all these things.
Mhm.
Yeah, so I guess a lot of
Yeah.
that.
How do you keep up with just, the latest advancements? Do you guys do journal clubs or, talk about it in, the cafeteria or the office or something like
Yeah, we definitely discuss papers quite often, especially when new ones come out. There's this preprint server by Archive where you can get like notifications from, so you know when like the freshest research has dropped. You
Yeah.
can also just like look on journal websites. But honestly, to answer your question, like Twitter is like the key place to be for. I guess it's X now, but used to be Twitter.
Yeah.
The key place for science. Like all the important papers that drop or all the new computational methods, they're all like dropped with like a thread and some links on Twitter, and then people comment and there's discussions. And I think that's like the greatest way to find literature at the moment.
Yeah. It's definitely like, at the palm of your hands too, right? When you're on your phone, it's so great.
Yeah.
So honest question, do you think there are too many epigenetic clocks out there? I think
Uh,
I appreciate that feedback.
yeah, there's so we actually try to get around this problem, or not necessarily get around the problem, but make it easier for people to use these clocks. My colleague in Vadim's lab, Albert, put out this preprint called Clockbase, and it's associated with a website, and you can just go on it and you can browse any data that's been uploaded to Geo, and the computations have already been performed for the epigenetic age clocks, or you can add your own data and just kind of get a nice data frame of your predictions. So hopefully it's easier for people to use it. It's kind of the inception. But yeah, I mean, they all look at different things, right? So like the Horvath clock was a multi-tissue clock. That the newest Horvath like mammalian clock tracks changes that are common between mammals, so perhaps they're a little bit different than just those in humans. You can have tissue-specific clocks, which are also useful, right? So you don't want to apply a clock train on blood to just the liver, and vice versa. So yes and no. I mean, I think it's a very promising area of research, clocks in general, including like transcriptomic and more of these single cell clocks. I'm hoping. At the same time, I think there's a fundamental truth about clocks, and that and there was one from True Diagnostic too that just came out last week as well. Omic MH.
Yeah.
It's really like the most accurate, and they use like electronic health records and
Mhm.
just biobank to date as well. So yeah, they're great over there. But it's still unclear kind of how these things translate over to function. Right? Same with like doing any kind of cell work that we do here. So okay, like maybe your therapeutic reduces the biological age of the cell or a sample by 2 years, 3 years, but does it actually make the cells do anything better, right? And like measuring that, I think, is crucial. So a lot of value to computational approaches. Ultimately, you need to validate with function, and it's harder in some ways and in some cases than others. So for T cells, for example, it's a much more established kind of the main assays that you would do to evaluate function. You co-culture them with tumors and you see if they kill tumors, right? That's like the major one. But there are some other cells where it's a bit more unclear how to properly measure their function and whether or not your rejuvenation therapeutic, beyond just shifting a number down in a graph, actually makes a meaningful difference in the body.
So do you think so like cells, single cells is one thing, but then I guess compartments, right? Cellular organelles. Like how's
Mm,
that, I mean, a lot of people are very specific on, how to tackle the mitochondria issue, right? Or like lysosomes now too, and like the ER basically. Um, but
yeah,
yeah,
it's getting more and more interesting because you're getting more methods to profile things at a
yeah,
crazier resolution than ever before. So
yeah,
there's now methods to do like subcellular profiling, and it's going to grow at scale, I imagine, as well. So as you're saying, I think, yeah, people right now are just kind of looking at, I don't know, some people are like very specific and just look at like mitochondrial aging, right? Or like ribosome aging or things like that. And then there's other kind of approaches that are very broad, but then that miss kind of the little things that are happening. And I think kind of integrating both is crucial. So like the systems level understanding of biology to where you make these like key discoveries about what's happening at the very localized level, like what changes occur with aging, but also kind of learning how to model them as part of a whole. Because eventually one would want a model of a cell or a model of an organism to model exactly what's happening with aging, right? Maybe it's just stochastic noise. There have been some preprints recently that are arguing that. Maybe there's some developmental aspects of it. It's just continued. It's still very unclear, right? And so being able to measure things and then somehow model them
yeah.
is
That's why I really appreciate longitudinal data, right? Because a lot of studies are just looking at one time point, but if you can track it, right? Because I think with cancer and Alzheimer's too, someone like one person is the risk factor of developing that. It peaks at like 60 at age 60 or something, and then comes back down, right? And it's really interesting to see how the aging landscape plays a role in that and what drives this increased susceptibility at that age. So
Good. Yeah, I'm all about longitudinal studies as well. I think they probably had some in this newest clock, this Omic
mhm.
MH, I suspect. But yeah, whenever you can do a longitudinal experiment, it's the most interesting. If you have your starting population and you can measure it, and then you see how it changes over time, even when you're reprogramming, right? You take
Yeah,
samples every day or every 2 days, or maybe with an automated system, you take samples every 3 hours, and you have a beautiful high resolution time course of how things are going.
yeah,
And
exactly. Well, I can spend all day picking your brain about this. But just one last question. What do you like to do outside of science? Do you have any hobbies?
ooh, indeed.
That's good to hear.
Yeah, I play the piano.
Oh,
Actually,
okay.
sitting next to my piano right now. It's right here. It's a beauty.
Oh, nice. Look like
Yeah,
you have a lot of music notes or sheets.
yeah. I have a lot of sheet music, a lot of records as well. Been collecting around music. But yeah,
Okay.
over the last couple of years, definitely like really into music. Ever since I started college, really. I had a couple of friends, including one, my friend Evan, who's a producer, and so he kind of introduced me to like
Yeah,
really appreciating music and just like the layers that go into it and how you kind of put it together. So I've been listening to a lot of music the last 7 years. Yeah, I love to hike as well. I live in the hills now, so there's a bunch of beautiful places to hike. There's a park nearby where there's just horses, so you can just go and walk with the horses.
yeah,
Yeah, things like that, and then just like, yeah, chilling with people, reading.
yeah. Do you
Also,
miss do you miss the beaches in France?
yeah, so I have very faint memories of the beaches
Oh,
in France. I was really young when we lived in the South, although my parents recently moved back to the South, so maybe I'll be able to go back to a beach, but not really. I would say, like, I think California is a pretty optimal place to live,
yeah,
in terms of the
yeah,
the mountains, the beaches, like the ocean, like
yeah.
everything.
The redwoods. Yeah,
Yeah, yeah, exactly. The trees.
yeah.
Yeah,
Cool.
beautiful place.
Okay, well, I think that's all we have time for. This was really nice and engaging. I appreciate you coming on to the
Yeah,
Scientific
yeah.
Spotlight. Um, and I guess if anyone had any questions, are you active on LinkedIn or Twitter or X? How would one reach out to you?
Yeah, I think X is a great one, or
Okay.
Twitter. I'm gonna have a hard time calling it X. It's
Yeah.
been Twitter for me for a while, but yeah, that works. I also have LinkedIn. Yeah, wherever people can reach me.
Okay. It seems
If
like
people
X. Yeah.
if people are interested in positions at Retro, we're always hiring for diverse positions, so you can check out our website as well.
Okay. Yeah. Well, thank you for joining us today. And to everyone else,
I appreciate it.
yeah, and to everyone else, thank you for watching and stay tuned for more scientific spotlights.